So hello everyone. So today we are going to talk about the Golgi apparatus. The Golgi apparatus consists of sacs. So these are flattened sacs, and these are identical to the, the cisternae what we have seen in the ER. But the difference between the cisternae of ER and those of the Golgi apparatus is that in case of Golgi apparatus, these cisternae are not physically connected to each other. So each one um, is a separate structure. The main function of the Golgi apparatus is to receive products from the ER, sort them, tag them, and then dispatch them. So these are four basic functions of the Golgi apparatus. It receives products from the ER. It will sort them into different groups. It will tag them for transportation to the destination. So tagging is more like a zip code, or like when you're shipping something to a destination. So it's more like tagging is more like an address where that product needs to be transported, and that's the fourth function, transportation. So receiving, sorting, tagging, and transportation, these are four different functions of the Golgi apparatus. The Golgi apparatus contains this has got two phases. There's both structural as well as functional directionality in the Golgi apparatus. So this is the cis end and this is the trans end, and things move from the cis phase, which is the receiving side of the Golgi apparatus, towards the trans phase of the Golgi apparatus, which is the shipping side of the Golgi apparatus. So Golgi apparatus would receive products from the ER from the cis end, and it will transport them or ship them off from the trans phase to either to other organelles or towards the plasma membrane. And sometimes these vesicles can also move backwards to the previous cisternae, or even backwards towards the Golgi apparatus. We will discuss these things later. So there are two models for the transpiration or for the movement of products from the cis phase towards the trans phase of the Golgi apparatus. One is known as vesicular transport model, and the second one is known as cisternal maturation model. The first model proposes, so these are two different models. There's kind of discussion or debate whether things move in vesicular transport model fashion, or the proteins move in a cisternal maturation model fashion. So what does vesicular transport model state? This model states that here you have got the ER. ER produces vesicles from the vesicles which contain different types of proteins. These vesicles fuse together to form this tubular cluster. This cluster then becomes part of the Golgi apparatus. But after it becomes part of the Golgi apparatus, these vesicles bud off from the this what we call the cis network, and then we move towards the second cisterna. From the second cisterna, proteins move in the form of vesicles to the third one, and from the third one in the form of vesicles to the fourth one, and then to the trans Golgi network, and later they will be dispatched in the form of vesicles from the trans end of the Golgi network. During this process, some proteins can also be transported back towards the previous cisternae. And the reason why they move backwards towards the previous cisternae is sometimes they're not properly matured. So the entire maturation process takes place inside the Golgi apparatus. When proteins are received from the ER in the Golgi apparatus, during their movement from the cis end towards the trans end, these proteins are matured. So different types of processes take place here, like we have discussed: sorting, tagging, and then transportation. So sorting and tagging takes place inside the Golgi apparatus during their movement from the cis end towards the trans end. But sometimes uh, if this vesicle moves from the cis phase towards the first or second cisterna, um, the protein is not properly matured, but it is again moved towards the third one. So when this is not properly matured, this protein is sent back to the previous sac so that it gets matured again. And this happens with the proteins in all the cisternae. So they can move back and forth in the form of vesicles during their passage from the cis end of the Golgi towards the trans end. So proteins are transported in the vesicles from the cis phase all the way towards the trans phase, and so that they can get matured. Sometimes these vesicles also move backwards towards the ER. So these vesicles could be carrying either the proteins, which were not yet properly prepared for the Golgi apparatus, or they move backwards towards the ER, because these proteins could be needed in the ER. So this is what we call vesicular transport model. It states that that proteins move from the cis phase of the Golgi apparatus toward the trans phase in the form of vesicles. That's why this is known as vesicular transport model. Another model states that this is not the case, but rather what happens that ER produces these vesicles. These vesicles fused together to form a vesicular tubular cluster. So this is the cluster, and this cluster fuses with the Golgi apparatus, and this becomes the cis Golgi network. Now, rather than the vesicles moving from the cis end towards the trans end, rather the entire tubular cluster, this is shifted in the Golgi towards the trans phase, or towards the medial region, and then from the medial region towards the trans phase of the Golgi. So this cluster moves to the second place, and from the second place to the third place, from third to fourth, and so on, all the way towards the trans end. So there is no movement of vesicles from one cisterna to the other one. It's rather the entire cisterna which moves, taking along all the content that it contains from the cis phase to the trans phase. However, during this process, proteins can move backwards to the previous cisterna in the form of vesicles. So there's no vesicular movement in the forward direction from the cis end towards the trans end, but proteins can move from the in the backward direction in the form of vesicles. So this is what we call cisternal maturation model. It means that the whole cisterna gets matured during its movement from the cis phase of the Golgi towards the trans phase. So all the proteins that this cisterna contains, they're matured. Some sort of tagging or post-translational modifications take place here, and then this cisterna is moved to the second place, and the first place is filled up by another vesicular tubular cluster, which is formed by the fusion of the vesicles which were ejaculated from the ER, and so on. And in this way, these cisternae move from the cis phase towards the trans phase, and during the movement of these cisternae, the proteins that they contain, they get matured, and later at the trans end, these vesicles, they bud off from the trans end, and they're transported towards either towards the plasma membrane or towards different cellular organelles. So these are two models. So proteins can either move through the Golgi apparatus in the form of vesicles, or they can move from the ER through the Golgi from the cis phase towards the trans phase in the form of as a cisternal maturation model. Let's talk about the movement of vesicles between the ER and the Golgi apparatus. How do they move from the ER towards the Golgi apparatus, and how do they fuse with the Golgi apparatus? So basically, this is the first step, how these vesicles move or they bud up from the ER, and then they fuse with the cis phase of the Golgi network. How does this movement take place? So that's what we are going to talk about. And then this backward movement. So we are basically talking about the movement of the vesicles between the ER and the cis phase of the Golgi. How do they move from the ER towards the cis phase of the Golgi, and from the Golgi back towards the ER? So this process is basically facilitated by some coat proteins. So these coat proteins, when they take this vesicle from the ER towards the cis Golgi network, these coat proteins are known as COP2 vesicles. So these are COP2 proteins. Basically, these vesicles are known as COP2 vesicles because they're coated with COP2 proteins. These COP2 proteins are basically coat proteins. So this is the rough endoplasmic reticulum, and as you can see, there are two types of proteins. There are either soluble proteins. These are cargo, means these proteins need to be transported somewhere, and then there are other proteins which are associated with some sort of membrane, transmembrane protein, or some sort of receptor. So they're either soluble cargos or they're membrane cargos. So both of them, the soluble cargos, they're received by the receptors in the membrane, and then there are membrane cargos which remain embedded into the membrane of the vesicle, which buds off from the rough ER. And basically this process is facilitated by the interaction with the COP proteins. As you can see that these proteins of the membrane cargo, they're interacting with the coat proteins, which are COP2 proteins in this case. So this vesicle buds off from the ER. This is coated with these proteins, and this vesicle moves towards the Golgi network. So as soon as it reaches closer to the Golgi network, these COP2 proteins, these are removed. They get dissociated from this vesicle. Now this vesicle contains the cargo proteins, which were soluble cargos, means they would be released. They're soluble, they're not part of the membrane, and it contains some membrane bound cargos, and it also contains these yellow in color, these proteins. So basically these proteins, which are yellow in color, indicated here in yellow, these are known as snare proteins. There are two types of snare proteins, V snare and T snare. So V stands for vesicular membrane, means the snare proteins which are located on the surface of this vesicle. And there are T snares. So T stands for target membrane. So T snares means those proteins which are present on the target membrane, where the vesicle has to land on, where the vesicle has to attach. So basically these proteins, the snare proteins, the V snares, which are found on the surface of the vesicle, and the T snares, which are found on the surface of the target membrane, both of these two proteins help in tethering process or in docking process, or in the fusion. They help in the fusion of the proteins to the membrane of the Golgi apparatus. So the V snare docking takes place between the V snares and the T snares. V snares are located on the membrane of the vesicle, while T snares are located on the membrane of the on the target membrane, or on the membrane of the Golgi apparatus. So after this docking takes place, the content inside the vesicle are released into the Golgi apparatus. So this was the forward movement or anterograde movement from the ER towards the Golgi. And as we have previously discussed, that some proteins can also be sent back to the ER. In both the cases, either they were missorted proteins, so they're sent back to the ER. They were not properly matured, so they're sent back to the ER. Some work was still needed to be done on these proteins, which could be done inside the ER. Or on the other hand, these could be the proteins which were needed in the ER. These were some sort of enzymes which were to be utilized inside the ER. So they're sent back from the Golgi back into the ER so that they can perform their function. These proteins were sent to the Golgi network for some sorting and tagging or some sort of post-translational modifications. And once they become mature, they are sent back towards the ER because they're needed inside the ER. So this backward movement also takes place in the form of vesicles, but this time these vesicles are coated with another type of protein, what we call COP1 proteins. So this is the reason these vesicles are also known as COP1 vesicles. And these vesicles again contain these snare proteins, and the membrane of the ER also contains snare proteins, and then docking takes place between these two snare proteins, and then this vesicle would fuse with the membrane of the ER and would become part of the ER. So we have seen that there are two phenomena. One is anterograde, or forward movement of the vesicles from the ER towards the Golgi apparatus, and the second one is the reverse, or retrograde transport from the Golgi apparatus towards the ER. What is important to remember is that in anterograde fashion, in anterograde or forward movement of the vesicles from the ER towards the Golgi apparatus, these vesicles are coated with COP2 proteins, while on their way back towards the ER, they're coated with COP1 proteins. And snare proteins are of two types: V snares, which are present on the vesicles, and T snares, which are present on the target membrane. These are two different proteins, but the job of both of these proteins is to help with the docking process between the vesicle and the target membrane. So these snare proteins help in the docking of the vesicles towards the membrane, whether it is the Golgi network membrane or it is the ER membrane. These vesicles are going to dock with both of these two membranes with the help of snare proteins. And this process is mediated by RAB proteins. So RAB proteins are basically, these are RAB GTPases. These are small proteins which help in the trafficking or movement of the vesicles from one place to another place. So we are basically, on the next slide, we will be looking at a slight more detailed phenomenon, or slightly detailed. We have this. How do these vesicles dock with this with the target membrane? So this is slightly more elaborated on the next slide. Let's see. Here we have got this vesicle. So this is our vesicle, and this is a membrane receptor, and this contains this is associated with the receptor is one soluble protein. Now this vesicle contains a snare protein, a V snare, because this is the snare protein associated with the vesicle. So this membrane contains, the membrane of the vesicle contains a V snare protein, and it also contains a RAB GTPase. Now RAB GTPase basically helps with the docking process. How would this do this? So the first step in this case is the tethering process. So tethering takes place between the RAB effector, between the RAB GTPases, which are located on the membrane of the vesicle, and the effector proteins or tethering proteins. These are RAB effector or tethering proteins. So these are the proteins which are located on the membrane, on the target membrane. So the vesicle basically, when it comes closer to the target membrane, this is encountered by the effector protein, RAB effector protein. So interaction between the RAB GTPases and the effector proteins is the first step that takes place here, even before the snare could form an attachment with the T snare. So when vesicle gets associated with this RAB effector protein, tethering takes place between the vesicle and this protein, the effector protein. Now these effector proteins pull this vesicle closer towards the membrane. They pull it closer towards the membrane. When they pull it closer towards the membrane, then docking takes place. That's the second step. So the first step is tethering. Tethering takes place between RAB GTPases and RAB effector proteins. So tethering means the vesicle is now attached via this anchor onto the target membrane. It's not yet fused with the target membrane, but it lies closer to the target membrane, and the effector protein here works more like a hook which is anchored onto this vesicle. Now the vesicle is fused with this hook-like structure. Now this effector protein pulls, excuse me, now this effector protein pulls this vesicle closer to the target membrane. When the vesicle becomes closer to the target membrane also contains snare proteins, what we call T-snares. Now docking takes place between the V-snare on the plasma on the membrane of the vesicle and the T-snares which are located on the target membrane. Once this docking takes place, this is followed by fusion of the membranes between the membrane of the vesicle and the target membrane, which could in this case be the membrane of the Golgi Apparatus, cis Golgi. So after fusion, the cargo or the soluble proteins they're released inside the Golgi Apparatus. So this is how basically vesicles mediate protein trafficking between the ER and the cis Golgi. So this is basically the detailed or elaborated structure for the docking of the vesicles onto their target membranes. So you got a vesicle which contains the V-snare proteins, and it contains Rab GTPases, while the target membrane contains T-snares and Rab effectors. So two proteins are here and two proteins are here. So first step, that is tethering, it takes place between the Rab GTPases on the vesicle and the effector protein on the target membrane. So that's the first step, tethering. This is followed by docking. Docking takes place between V-snare and T-snare. And the third step is fusion, where the membranes of the two structures, they are fused together and the soluble content is released inside the membrane, while the vesicle itself becomes part of the membrane of the Golgi Apparatus. And the membrane associated proteins, they stay embedded into the protein. So let's talk about some coat proteins. So we have previously discussed that we have got these. We have previously talked about these two types of coat proteins, that is COP2 vesicles and COP1 vesicles. We have basically three different major coat proteins are involved in vesicular trafficking inside the cell. And the first one is COP2 vesicles, but we have already discussed that for an anterograde movement or a forward movement from the ER towards the Golgi Apparatus, the vesicles are coated with COP2 proteins, and on their way back they are coated with, or in a retrograde fashion or backward movement, they're coated with COP1 proteins. Another type of protein is known as clathrin. When proteins move from the trans end either towards the plasma membrane or towards the endosomes, these vesicles are coated with clathrin, another type of protein. And this clathrin also coats the membrane pits, the pits on the plasma membrane. These are also coated with clathrin proteins. And when endocytosis takes place, then this vesicle, which is the result of endocytosis, this vesicle is coated with clathrin. And then both of these two vesicles, the clathrin mediate vesicles coming from the Golgi Apparatus and the clathrin coated vesicles, which are result of endocytosis, they can both fuse together to form an endosome, and later the lysosomes can also fuse with these endosomes. So these are three different major coat proteins which are involved in vesicular trafficking: COP2 vesicles, COP2 proteins, COP1 proteins, and clathrin. Okay, so that is sufficient for today, I think. Thank you very much for your time and understanding. If you have any questions, you can post these questions, and I would be glad to answer your questions. Thank you very much.